thank you very much, James, for agreeing to give a Build a Cell seminar, and it's all yours.
Share the screen. Is my screen visible?
It's working on it.
Great.
Yep, now it's visible. Thank you.
Great. Thank you so much. Thank you so much, Kate, and thank you all for the opportunity to share our work on the process of cell division in a genomically minimal cell. And I very much admire the research of the Build a Cell community, and I'm very much looking forward to your feedback and the opportunity to discuss with you. I'll be presenting on behalf of this collaboration between a team at the JCVI led by John Glass, Elizabeth Strakowski at NIST, Andreas Merschen, Neil Gershenfeld, and I at the MIT Center for Bits and Atoms, and Mark Ellisman and Tom Deerning at UC San Diego. And I'll describe two projects. The first project has been published, and in this project we determined which genes are required for a genomically minimal cell to divide normally. And some of these genes, the molecular mechanism of the gene is not known, how it contributes to normal cell division. So that leads us to the second project, which is ongoing, in which we're applying physical models for vesicles to the process of cell division in this organism to gain some intuition and generate hypotheses for how genes might contribute to cell division in this genomically minimal context. So for a background for the talk, and also a personal background for myself, there are different approaches to building a cell. And the wide range of talks in the Build a Cell seminar series cover the wide range of approaches to building a cell. And there's a class of approaches called bottom-up approaches, in which the macromolecular building blocks of cells, including proteins, nucleic acids, and membranes, are isolated either from a living cell or synthesized. And then these components can be mixed and matched and combined to reconstitute artificial cells that have some of the functionality of living cells. And a major advantage of these bottom-up approaches is that one can turn biophysical and biochemical knobs that are hard or even impossible to turn inside of a living cell. And a limitation of this approach is that currently it's not yet been possible to reconstitute a fully living cell from these non-living components. Although this approach, as it's based on the building blocks of cells, does allow one to study the physical properties and the chemical properties of these components to learn how they might behave inside of a living cell. My very first experience in research was in the lab of Seok-Jun Jun, where we isolated chromosomes from E. coli to study the physical properties of the chromosomes. And we showed that the chromosomes behave as soft springs that expand rapidly upon lysis of the cell. And then in the presence of a crowding agent, in this case polyethylene glycol, they rapidly compact back to a size comparable to the in vivo size. So this helps us gain physical intuition for how the chromosome might behave inside the cell and the forces that are relevant to the chromosome. And part of my work as a graduate student was based in the labs of Timothy Mitchison and Nikta Fakhri. Timothy Mitchison was my thesis supervisor. And we were working with cytoplasmic extracts from Xenopus laevis frog eggs. So this is a classic model system for studying the eukaryotic cytoplasm and the mechanics for eukaryotic cytoplasmic networks, such as microtubules, filamentous actin, as well as membrane networks such as endoplasmic reticulum and mitochondria. And so in this project, also within the bottom-up class, we were studying the mechanical properties of these networks. And in particular, we were looking at how these networks help position the division plane as well as move nuclei inside the cell. Another major class of approaches to building a cell is the top-down approach. So in this case, starting from a living cell, one attempts to eliminate as many genes as possible that are not required for growth in a controlled laboratory environment, resulting in a cell that has just a minimal gene set. And also as a graduate student, I had a chance to collaborate with Elizabeth Strakowski, John Glass, and Neil Gershenfeld to study the mechanics of cell division in these genomically minimal cells, and that's the project I'll be describing today. And I've just started a postdoc in the group of Saul Aries in the group at the National Center for Biotechnology in Madrid, and I'll be studying similar themes of cellular mechanics in biology. So for background on the minimal cell, JCVI Syn3A, developed at the JCVI, minimal cells are enabling fundamental studies on the basic principles of life. So this is a representation of the genome of JCVI Syn3A. It contains just less than 500 genes total, and less than 100 of those genes currently do not have a known function. And there are dozens of groups around the world, many of whom are participating in the Build-A-Cell community, who are studying the cell from different angles. And the number of genes of unknown function is decreasing, and a major goal for the community is to attribute functions to all of the genes to develop an increasingly complete model for how the cell works. In comparison, this is a representation of the genome of E. coli at the same scale, which contains about 10 times as many genes and about 20 times as many genes of unknown function. So E. coli is a great model system as well, and this is for comparison depending on the question. The small number of genes in JCVI Syn3A can be advantageous. So those genes are then expressed into gene products, and that's a function, of course, not only of the genome, but also the environment in which the cell is, as well as the history of the cell and its environment. So here's a representation of the proteome of JCVI Syn3A, broken up into essentiality. And a key point of this proteomic data is that a fraction of its essential proteome does not have a known function. So those genes that remain of unclear function, it's not that they're just not expressed. They're expressed and they're doing something inside the cell that's necessary for the cell to live. So this highlights a great amount that remains to be discovered in this model system. And a major goal is to how do you relate these gene products, hundreds of gene products, to the complex structure and the dynamic structure and the function and the phenotype of this cell. This is an illustration of JCVI Syn3A that was recently published by David Goodsell. And one can appreciate how complex is the structure in terms of the diversity of building blocks that it's made up of. And here we're studying the process of cell division, the phenotype of cell division. And the theme of this talk is, rather than go straight from genes to the function or the process of cell division, to consider mechanics, cellular mechanics, as an interlayer between genotype and phenotype. So in particular, we're breaking the question up and asking which values of these small number of physical parameters, the volume to surface area ratio and the preferred membrane curvature, can give rise to the process of cell division. And then in turn, how do these physical parameters depend on different genes that are present in this genomically minimal context? And just to be clear, by mechanics, I mean not molecular mechanism, but rather forces, bending rigidities, and viscosities at the scale of the cell, kind of blurring our eyes and taking a more coarse-grained look at how the cell behaves. So an outline for the remainder of the talk. I'll first summarize some technologies for building cells with synthetic genomes that are relevant to this talk. And then I'll introduce the genomically minimal cell JCVI Syn3A, and then describe the development of normal cell division in this cell. And our motivations were to provide a platform that's more controlled at the cellular scale. And also, cell division is an example of a phenotype or a process that has a complex genetic basis with multiple genes contributing. So this work illustrates, or rather provides an example of engineering this complex phenotype. There are other complex processes that likewise depend on a wide range of genes. So this is an example of that. And then the last part of the talk will be working to understand cell division by applying models that have been developed for vesicles. So 3 technological steps that were developed at the JCVI and elsewhere. The first is genome synthesis. So it's possible to synthesize whole bacterial genomes starting with oligonucleotides. And a key point that I'll highlight here is that before the genome is in the bacterium, it exists in yeast as a centromeric plasmid. And it's largely an inert piece of DNA inside of the yeast. So it's possible to use yeast genetics to edit this bacterial genome. And it's also possible to make large edits because there's not a requirement for viability at every step in the genome engineering process. So then how does one take that genome from the yeast nucleus and then boot it up inside of a living cell? So there's a process called genome transplantation that was developed at the JCVI Syn3A. And these projects I did not contribute to. I'm summarizing work that was done previously. So here, this is a representation of a yeast cell. A donor genome is isolated from that yeast cell and then combined with a cell of a related mycoplasma species. And then by a technique which is similar to polyethylene glycol and calcium-based chemical transformation, that donor genome arrives into the recipient cell cytoplasm. And the recipient cell lacks enzymes that can perform homologous recombination, so the two genomes are, in a sense, insulated from one another. And as only the donor genome confers resistance to an antibiotic, over time, only the donor genome is left, and the cytoplasm of the recipient cell, within 10 or more generations, is entirely encoded by the genes from the donor genome. So in this way, a donor genome can reprogram the recipient cell, resulting in a cell that is encoded by that donor genome. And then these previous technologies can be integrated into design build test cycles in which genome synthesis and genome transplantation is used to generate cells. And these design build test cycles were used to eliminate non-essential genes and iteratively decrease the genome size, resulting in a cell that had predominantly essential genes. So with that, I'll describe development of this genomically minimal cell at JCVI Syn3A. So Syn3A, its genome sequence is ultimately derived from a bacterium that exists in nature called Mycoplasma mycoides, and here we're working with the subspecies Capri. And this is an opportunistic pathogen of goats. And as a pathogen, it lives inside of a host organism. And as a result, it imports complex metabolites from the host. And as it doesn't require the genes to synthesize those metabolites, they can have naturally small genomes. So that's advantageous for developing a minimal genome because you're starting from an already small starting point. But the flip side of that coin is that they require a complex growth medium that includes nucleotides, complex lipids, amino acids, etc. So having a small genome requires a complex growth medium. And another key point about the mechanical properties of this cell, which is important to this talk, is that unlike many bacteria which have a peptidoglycan cell wall, Mycoplasma mycoides do not have a cell wall. Some mycoplasmas have a polysaccharide capsule, but it's not cross-linked to the extent that peptidoglycan is. And then JCVI Syn 1.0, using the genome synthesis technology, largely mirrors the natural sequence of Mycoplasma mycoides. And then using these design build test cycles, JCVI Syn 3.0 represents a subset of the genes contained in Syn 1.0. And it's called Syn 3.0 because during the process of genome minimization, there was a 2.0. And it's a challenge to minimize these genomes due to something called synthetic lethality. So the idea is that multiple genes could encode an essential function, and the process of identifying genes that are not essential often goes one at a time. So if one knocks out these genes that are individually not essential but together essential, then the cell is no longer viable. So the JCVI leveraged a kind of modular approach where the genome was assembled from 8 segments, and then different segments were minimized independently and then mixed and matched. So after this iterative process, this small group of this group of genes was left, and the details of that work are described in this paper here. And then JCVI Syn3A contains more genes than Syn 3.0. And in particular, Syn 1.0 has a normal morphology, similar to the wild-type Mycoplasma mycoides, and that there are many cells that are small and on the order of a micron or smaller. In scanning electron micrographs, about 400 nanometers is the estimate for the radius, for the diameter, apologies. And they're largely separated from one another, consistent with normal cell division. Syn 3.0, on the other hand, forms these large clumps and aggregates, including large cellular structures. So Syn 3.0, though it grows exponentially at the population scale, it's lost the ability to divide normally, similar to the wild type. So it's lost control of its morphology. And it's advantageous to have a platform which is controlled and less variable at the single cell level for engineering the cell and for understanding the physiology of the cell. So that was a goal for the development of JCVI Syn3A. And during the work that I'll describe next, we identified which genes are required for normal cell division. And Syn3A, though it doesn't look exactly like the wild type cell, it doesn't exhibit these large cellular aggregates, and for the most part, it includes cells that are submicron, similar in appearance to JCVI Syn 1.0. This work, development of normal cell division, was recently published, and I'll describe that work now. So these are higher resolution views of Syn 1.0 and Syn 3.0, and note that these scanning electron micrographs are shown at the same scale. So Syn 1.0, there's very low variability in cell size and shape, whereas in Syn 3.0, some of the cells are multiple microns in diameter. They're largely connected together. So looking at these static images, a question motivating this work was, how does Syn 3.0 self-replicate dynamically? What are movies to go along with these static images? And furthermore, which genes are required so that Syn 3.0 could recover the normal morphology and normal cell division exhibited by the wild type? So we adapted a microfluidic chemostat to image the growth of bacteria. And this was based on earlier work that I contributed to in the lab of Seokjun Jun, where we developed these rod-shaped channels. The Jun lab developed these rod-shaped channels, and the E. coli have the same width as the channel. And they are loaded into this channel, and there's a continuous flow of fresh growth medium across the open face of this channel. And the channel is about 25 microns long. And there's a continuous diffusion of nutrients into the growth channel, which is fast due to the small size of the channel. And so the cells are growing in a quasi-steady state, fed by the continuous flow of fresh nutrients. And as cells grow, they are displaced out of the channel. But as the mycoplasmas that we're working with are different sizes and shapes, as opposed to the rod shape of E. coli, we just adapted the design of this device, and we created kind of pancake-shaped channels, but based on a similar principle of diffusive buffer exchange. One reason this is important is because the mycoplasmas lack a peptidoglycan cell wall, and so they're sensitive to shear flow, where flow can deform the cells and could even break them apart. And so as the cells are growing in these chambers, they're shielded from flow, and as a result, the morphological dynamics that we observe should result from the intrinsic forces inside the cell, as well as confinement within the chamber. And here's an example of these pleomorphic cells growing inside of a microfluidic chemostat, and these movies are shown at the same scale. In contrast, as these cells grow, they fill up the chamber. So these cells are in less of a steady state than these because the density of cells is increasing in the chamber over time. But still, diffusive buffer exchange is helping maintain constant, or is helping to contribute to replenishing the growth medium inside the chambers. So here's a movie that shows a control showing the growth of JCVI Syn 1.0 in these microfluidic chambers. So the top row is phase contrast, and in the bottom row, the cells are constitutively expressing M. cherry in the cytoplasm. And the cells are small and near the optical resolution of the microscope, and they're rapidly diffusing around inside these microfluidic chambers. And here's a movie showing the growth of JCVI Syn 3.0, which in the microfluidic hemostat as well exhibits this abnormal division resulting in morphological variation, where there are some cells that are large, other cells that are small, some filamentous cells, and there's a variety of sizes and shapes that are observed. And the chemostat also allowed us to investigate not only the shape and the size and shape variation, but also the compositional variation by using different fluorescent dyes. So here we're looking at cytoplasm stained by just constitutively expressed M-cherry. Some cellular structures contain a marker for cytoplasm, and then other structures did not contain cytoplasm, though they excluded a fluorescently labeled dextran from the growth medium, suggesting that this structure here is impermeable to the dextran, though it doesn't contain cytoplasm. So we don't know what those structures are. We've been referring to them as vesicles generated by the cells. This suggests that dysregulation of membrane accumulation is somehow related to this phenotype of abnormal cell division, but we don't understand the details, and that's something that we're looking into. And then we also labeled the DNA with Hirsch to look at the distribution of DNA. And likewise, there's variation in the intensity of this label. We don't know how chromosome copy number varies, but abnormal division is likely correlated with abnormal chromosome segregation and variable numbers of gene copy numbers in the different cells. So which genes are involved in this process? So as noted, the Syn 3.0 genome is composed of these 1/8 segments that mirror the 1/8 segments of the Syn 1.0 genome. So it was possible to mix and match one minimized segment in the context of an otherwise wild-type genome. And so we screened all 8 minimized segments. And fortunately, only the minimized segment 6 conferred this abnormal cell-shaped phenotype. And interestingly, hundreds of other genes could be deleted, dozens at a time, and the cell size was still normal. So this helped restrict our search from the 428 genes that were missing in the Syn 3.0 genome to the 76 genes that are missing in the genome that has only the minimized segment 6. And the hypothesis is that some genes were missing, and then those genes would somehow contribute to normal cell division, which would result in a normal cell size and shape distribution. Although it is possible to imagine that overexpressing a certain gene could also mess up cell division, but that's something to be studied in future. So for now, we followed this phenotype, and we looked at the growth of this strain in a microfluidic hemostat, and as with Syn 3.0, the cells exhibited size and shape variation. And then, with our attention on the minimized segment 6, this strain, JCVI Syn 3A, was identified. So the JCVI, while minimizing the genome, they developed increasingly minimal genomes. And this was one strain that had been developed during the genome minimization process that was nearly minimal, but contained 19 genes that were not present in Syn 3.0. And this strain had not been imaged at the cellular scale. And this strain behaved similar to wild-type Mycoplasma mycoides in JCVI Syn 1.0, and that the majority of the cells were small and near the optical resolution. So here are the genes that are retained in Syn 3A that are not retained in Syn 3.0. And they were organized into 8 clusters of genes that are adjacent to one another. And because they are adjacent, they could represent transcriptional units. So we therefore first treated them as intact clusters, and then later on investigated the requirements for each gene individually, as well as a second copy of a ribosomal RNA operon that we showed was not necessary for normal cell division, so we didn't add a a label to that one. So we noticed that one of these clusters included the cell division protein FDSZ. This is highly conserved in bacteria. It's a tubulin homolog that forms filaments that localize at the division plane in many bacteria. This is an electron cryotomograph of an FDSZ ring constricting a liposome to illustrate it forms these rings, and these rings are implicated in cell division. And it's present within a highly conserved cluster of genes that are known to participate in cell division. So when we saw that FDSZ was present in JCVI Syn3A, that was actually one of the ways that JCVI Syn3A was identified was, oh, this is one that contains FDSZ, so let's look at that one. So we hypothesized, oh, maybe it's FDSZ that's missing and the cell needs FDSZ to divide normally. But to our surprise, if just this cluster of 3 genes, including FDSZ, was added back to the genome of Syn 3.0, cell size and shape was still crazy. The cells did not divide normally and still exhibited this extreme morphological variation. So then we had to consider these other genes. And looking at what's currently known about the gene function, there were no major hints about what else might be happening. So this work was led by co-first author on this project, Liji Sun, at the JCVI. So Liji developed systematically a number of strains to test hypotheses about different combinations of genes to find which combination of genes is the smallest number of genes necessary to confer normal cell division. And this was done using genome editing techniques, both in the yeast as well as after transplantation into the mycoplasmas. So after generating many strains, Liji identified that these 7 genes distributed across these 3 clusters are together necessary for normal cell division in this organism. And furthermore, Liji added these genes to an ectopic locus and then deleted them individually. And because the genes were added back not to their native genomic context, but rather somewhere else in the genome, this is some evidence that the endogenous genomic locus hasn't been perturbed, but rather it's the presence of these genes that confers normal cell division. And while FTSZ is known to participate in cell division, and this other protein, CEPF, is known to help FTSZ interact with the membrane, these other 5 genes, some of which are highly conserved in bacteria, including this gene 527, which has a highly conserved domain, as well as gene 604, which likewise has highly conserved domains and is part of a highly conserved family of proteins. The molecular mechanisms of these genes are unknown, and we don't know how they contribute to the process of normal cell division. So that brings me to the last part of the talk, in which we're applying models of vesicles to cell division in this organism. And here especially, so this is work in progress, and I've been studying this literature, and people who are present in this very audience are world experts on this topic. So I would be really delighted for your feedback, and I'm really appreciative for the chance to present this to you. So a question motivating this part is, what forces are driving cell constriction and cell scission during cell division in this genomically minimal context? And here, these shapes are adapted from models developed for vesicles, which in a way provide a simple starting point for membrane mechanics. So one can imagine a cell growing, both its volume and its surface area, and then constricting, and then eventually the cell completely dividing to form 2 separate units. And these are actual shapes predicted for vesicles in the absence of FTSZ or any specific protein. So as a proof of concept, vesicles can execute these division cycles, and that's been shown, and see this work in particular by Bozek and Svetina. And one would ask, how is this process controlled in JCVI Syn3A, which in addition to a lipid membrane contains a number of proteins that can act on that lipid membrane. So first question is, might models of vesicles apply to JCVI Syn3A? And we first observed that in the microfluidic chemostat, when we image not over timescales over which the cell is growing, over the minutes to tens of minutes timescale, but rather on the sub-second timescale, the cells exhibit these fluctuations. And unlike rod-shaped bacteria, the fluctuation just qualitatively, we haven't tried to quantify this yet. This is something we'd like to try. But just qualitatively, the fluctuations appear consistent with those of fluid membranes based on how they wiggle over short timescales. So in particular, we're focused on a model called the spontaneous curvature model, which is a simple model for vesicles, which has a small number of parameters. And here we'll argue why this model may apply to JCVI Syn3A. So the spontaneous curvature model makes two key assumptions, or two among other assumptions. So one is that vesicles have shapes that are in mechanical equilibrium. And the second is that flip-flop of at least one component between the two leaflets allows the area difference between the leaflets to change. So regarding this first assumption of might JCVI Syn3A have shapes that are in mechanical equilibrium, what are non-equilibrium processes that might cause the shape to be out of equilibrium? So the first we would consider is the process of actively growing. So this recent work by Ruiz Herrero et al. Has looked at a regime in which growth of the membrane is so fast that it's not able to mechanically equilibrate. And rather than having these smooth shapes that are in mechanical equilibrium, there are these highly wrinkled shapes where that excess surface area is taken up in these wrinkles. And the wrinkles have a typical size of length L. And this mechanical relaxation length scale is a function of the membrane bending rigidity, which reflects how hard is it to bend the membrane, the growth rate of the membrane, as well as the membrane viscosity. So the membrane bending rigidity has not been measured in Syn3A or other mycoplasmas to our knowledge, but a common order of magnitude for liposomes as well as biomembranes is 10 to the minus 19 joules. And the growth rate is a function of the doubling time. So JCVI doubles every 105 minutes roughly. And the growth rate is, one would say, okay, 2 to the t over tau, and then express that in terms of, if you were to say the area is increasing exponentially with this as e to the, I should have written this down, I apologize, as e to the gamma t. So this growth rate is the natural log of 2 divided by the doubling time. Please ask if there are questions about that. So that turns out to be 10 to the minus 4 per second. Anyways, this growth rate is, I'm more accustomed to thinking about growth rate in terms of doubling time, but it's possible to refer to it as in per second scale. And the third parameter, the membrane viscosity, that too hasn't been measured in JCVI Syn3A, but it has been estimated in Mycoplasma mycoides, from which Syn3A was derived. And the value of that is 10 to the minus 1 Pascal seconds. So combining these three parameters, we can estimate a length scale of 10 microns. So in other words, these wrinkles would appear in a Syn3A cell that had a typical size of about 10 microns. But as Syn3A is significantly smaller than that, we can argue that Syn3A is likely in this regime in which mechanical relaxation is much faster than growth. And so that's consistent with the first assumption of the spontaneous curvature model. There are other active processes that might be happening inside JCVI Syn3A that could also cause non-equilibrium shapes. So in particular, the Z ring is hydrolyzing GTP. I don't think that's been shown specifically in Syn3A, but that's been shown in a number of other model systems. And then furthermore, Syn3A retains ATP synthase, and this recent work has shown that active fluctuations driven by ATP synthase in liposomes can cause non-equilibrium shapes as well. So these could contribute, and this remains an area of future research. But for now, this being in this regime of fast mechanical relaxation with respect to growth is an argument why we'll apply the spontaneous curvature model. The second assumption relates to flip-flop of at least one component. And this work from the Shostak lab has shown that cholesterol can rapidly flip-flop between leaflets with a residence time of 10 milliseconds, which is much faster than the time scale associated with growth. So in particular, if you deform a bilayer, then one leaflet will become sparser and the other leaflet will become more compressed. And flip-flop of one component will allow the densities to equilibrate and will allow the stress to relax. And this recent mass spectrometry analysis from the Sayans lab has shown that cholesterol is the most abundant lipid component in JCVI SYN3A. So because JCVI SYN3A retains a lipid component that can rapidly flip-flop, we feel that the spontaneous curvature model may apply as well. So now to introduce the phase diagram for the spontaneous curvature model. One axis is the volume to surface area ratio, where this metric, the far right side of this phase diagram, corresponds to a fully inflated vesicle, in the sense that a sphere has the maximal volume given the surface area. And then the far left side of this axis corresponds to a fully deflated vesicle, which is all surface area and no volume. And this could have a wide variety of shapes. For simplicity, I've shown it as a flat structure, but it could have many different shapes. And then the vertical axis of this phase diagram is called the spontaneous curvature, which is a measure for the preferred membrane curvature that corresponds to zero bending energy. So the spontaneous curvature reflects local interactions, molecules that locally bias the curvature of the membrane. And there's a single parameter which describes the entire vesicle. So the assumption is that these curvature-inducing molecules are uniformly distributed over the vesicle surface. And positive spontaneous curvature corresponds to convex shapes, where the membrane tends to curve away from the inside of the cell towards the outside of the cell. And then negative spontaneous curvatures have a more concave shape, where the membrane prefers to curve towards the inside of the cell. And then higher magnitudes correspond to higher curvatures. And there's a kind of a tension or trade-off, not trade-off, but the spontaneous curvature is a local parameter, and the surface area to volume ratio is a global constraint. So if a vesicle is fully inflated, then its mean curvature is dictated by the surface area to volume ratio and by the vesicle size. Where if it's a sphere, and then the mean curvature is 1 divided by the radius of that sphere. And the spontaneous curvature parameter could be anything. So in other words, this could be held in a spherical shape due to its surface area to volume, although the membrane wants to bend due to these curvature-inducing molecules. And so it's this trade-off that gives rise to this complex phase diagram, where there's a number of shapes, including ellipsoidal shapes, stomatocytes, which include inclusions on the interior of the vesicle, as well as pear-shaped vesicles, which are constricted, as well as this red line corresponding to fully constricted spheres that are joined by an infinitesimally small neck connecting them. And just qualitatively, JCVI SYN 3.0 exhibits shapes that live in this phase diagram, including, this is based on wide field microscopy, so we don't have a good measure of the shapes in 3 dimension at the resolution of the cells, but qualitatively, there are spherical shapes, there are ellipsoidal shapes, there are filamentous shapes, and then this shape seems to have some kind of an inclusion. There are other shapes qualitatively similar, which are kind of in the perhaps in the stomatocyte region. So this is an argument why the spontaneous curvature model might apply to SYN 3.0. And then considering cells that divide normally. So we zoom into this top right region of the phase diagram, and that's shown here. And it's just replotted the same region of the phase diagram. So the far right boundary, as noted, corresponds to a single sphere. And then this left boundary corresponds to 2 spheres of equal size that are joined by an infinitesimally small neck. And trajectories from this line segment to this line segment correspond to possible shape trajectories over the cell cycle. So for a cell to fully constrict and divide symmetrically, then the volume to surface area ratio should decrease, and the spontaneous curvature should be sufficiently high. And so this model makes quantitative predictions for how JCVI Syn3A should control these biophysical parameters in order to divide normally. And just for intuition, where did these square roots of 2 come from? So one can ask, there's an equivalent radius, which is for the given surface area. Well, if this formed a sphere, what radius would that sphere be? So if we consider 2 spheres of equal size, each of which has radius r, if one were to reshape this vesicle into a single sphere, then it would have a equivalent radius of r naught, which is the radius of one sphere times the square root of 2. And that would involve inflating this vesicle and increasing its volume. So then this volume to area ratio is equal to the actual volume divided by the maximum volume. And here the actual volume is 2 times the area of each sphere, and the maximum volume is the volume of a sphere of radius r naught. And if one does the math, one gets 1 divided by the square root of 2, so that's this here. And then this dimensionless spontaneous curvature is the actual spontaneous curvature, which has units of 1 over length, times this equivalent radius. And the intuition here is that larger vesicles that are inflated would tend to have smaller curvatures. So the dimensionless curvature multiplies the actual spontaneous curvature times that equivalent radius to get a dimensionless parameter. So here, each subsphere has a curvature of 1 over r, and then the equivalent radius is r times root 2, and so this dimensionless curvature has a value of square root of 2. So this point in the phase diagram corresponds to these 2 spheres of equal size. And in addition to driving constriction, the spontaneous curvature model can predict scission driven by curvature. So in particular, this model considers how much energy would be required to open up some holes in the membrane, exposing the hydrophobic core of the bilayer, and then asks how much force could this constriction force generate, and then asks at what point can the energy released by constriction overcome the energy barrier required to open up these holes in the membrane. And so this recent work looks at how that constriction force scales with the spontaneous curvature here, m. And at sufficiently high spontaneous curvatures above this purple dotted line, there's predicted to be enough force to overcome that scission barrier. So now we shift the discussion to ask which genes in JCVI Syn3A might contribute most to these physical parameters. So in particular, of course, many genes are contributing to the cell volume and to the cell surface area. But we're focusing on which genes might have the most effect on volume or surface area. And in particular, water accounts for 65% of the volume of the parent organism, Mycoplasma mycoides. And so here we're considering what genes might JCVI Syn3A retain that are involved in osmoregulation and regulating the flow of water. So this is a representation of genes that are known to participate in osmoregulation in E. coli. And this include ion transporters, as well as transporters for organic metabolites, as well as aquaporins and these mechanosensitive channels. And very few of these genes are retained in JCVI Syn3A. The potassium transporter, TRKA, has been annotated. A glutamate transporter has also been annotated, and a choline transporter has not been annotated. However, choline has been shown to affect intracellular potassium levels in Mycoplasma mycoides. So we might speculate that Syn3A could retain a choline transporter. Syn3A currently retains 15 unannotated transporters comprising 22 proteins. So there remains a lot of work on these transporters and which might contribute to osmoregulation and modulation of the volume by affecting the osmotic pressure and the resulting transport of water. And then as far as transport of as far as growth of the surface area, we consider which genes might contribute most to incorporation of lipids. The mycoplasma membrane is comprised of approximately 50% lipid and 50% protein. So in a sense, every protein contributes to some fraction of the area. But here, there's a small number of lipid species. So which proteins regulate the incorporation of those lipid species? So there's a system that's been described in other bacteria, including fatty acid kinases in the system FAC-A and FAC-B. So fatty acids are phosphorylated upon incorporation into the membrane, and that's how the phospholipids are formed. So Syn3A retains these genes. So these genes might be of interest for future studies looking at regulation of the volume to surface area in this cell. And then the second parameter, the spontaneous curvature, which genes might contribute to that physical parameter? So we're going to note possible mechanisms that could generate sufficient spontaneous curvature to constrict the cell and possibly even divide the cell. And as the cell has a radius of about 200 nanometers, we're going to consider mechanisms that have been reported to generate curvatures that are higher than 1 divided by 200 nanometers. So the first, Syn3A retains the lipid cardiolipin, which is has a conical shape with an intrinsic curvature of 1 over a nanometer. And it retains a gene, a cardiolipin synthase, and cardiolipin has been shown to localize to curved regions of the bacterial membrane in other species. So future work could look at the role of cardiolipin during cell division in this bacterium. As a second class of models, as shown in this reference here, as well as in other work, if there are proteins that are in the outer leaflet, but not in the inner leaflet, crowding of these proteins in the outer leaflet can generate a kind of a pressure that causes the membrane to bend away to increase the space available to these proteins that are in the outer leaflet. So it's a kind of a crowding-induced curvature. And here we note that Syn3A retains about a dozen uncharacterized lipoproteins that would be displayed in the cell surface. And then looking at the proteomic data and the estimated surface area of Syn3A, we can estimate a total density of about 3,000 lipoproteins per square micron. And in this work in liposomes, 150 GFPs estimated per square micron gives a spontaneous curvature of about 1 divided by 250 nanometers. And so this is going to depend on the properties of these molecules, so it's not possible to compare these directly, but it seems that crowding of lipoproteins could possibly generate curvature that's relevant to this process of cell division. We previously noted that Syn3A retains FTSE, which polymerizes into cytoskeletal filaments. These interact with the membrane via proteins such as FSEPF and FTSA. And in vitro, these filaments can form curvatures that are 1 divided by several nanometers. In vivo, the dynamics of FTSE in Syn3A is not currently known. And then as a last possible mechanism that we're considering, lipids from the growth medium are likely incorporated into the outer leaflet. And then there's a flipase enzymes that hydrolyze ATP to pump lipids from the outer leaflet into the inner leaflet. So those are genes 371 and 372. And so depending on the rates at which lipids incorporate, lipids are pumped to the inner leaflet, and then cholesterol is moving across the leaflets as a function of the abundance of cholesterol. One can imagine an area difference between the leaflets potentially contributing to curvature. And that's not consistent with the spontaneous curvature model specifically, but there's an extension of the spontaneous curvature model called the area difference elasticity model. And this kind of effect could be considered within that extension of the spontaneous curvature model. So in summary, a key question motivating this project is, how do these genes, specifically the genes of unknown function, contribute to the process of cell division in this genomically minimal context? And currently we're considering mechanics as providing an interlayer in which we ask which values of these physical parameters, such as the volume to surface area ratio and the preferred membrane curvature, can generate cell division, and then which genes might these parameters depend on. And this is just a summary of the genes I listed in the previous slides. And then these are the 4 genes of unknown function that are known to be required for normal cell division. So these studying these models developed for vesicles has been inspiring in terms of thinking about these genes could potentially contribute to, say, regulation of membrane accumulation, or they could introduce curvature by FDSE independent mechanisms. And then just extrapolating, there's a number of other possible frameworks and conceptual frameworks between the full set of genes and the complex physiology of the cell, including thermodynamics and metabolism, information processing, a number of physiological laws in which one doesn't consider each gene or gene product separately, but rather proteome sectors such as ribosomes and non-ribosomes, or how might the cell have evolved, and how have the genomes of these reduced species evolved related to where they're found in nature. So with that, I'd like to thank you all again for the opportunity to present. Thank all the co-authors on these projects, as well as technical advice from Jason Kralish at NIST, and thank you again for the chance to present here.
Thank you very much, James. That was awesome.
Oh,
There is a
you.
lot of questions in chat. If you could read the question before you answer it, that's going to be helpful for people watching the recording.
Oh, absolutely. And I'm sorry I didn't see these as I went.
No, that's okay.
Oh, okay. So there was a question about the mother machine. That's right. That was the mother machine developed in Seokjun Jun's lab, and I had a chance to. It was my first research experience in Seokjun Jun's lab as an undergraduate, so I was learning from that group, and so it was so that was the background in that project. And currently there's a wide range of groups that are developing microfluidic hemostats to study different aspects of bacterial physiology. Okay, so a question from Azim. I'm going to expand this. It doesn't show the full name. I'm going to look at the participants list. Azim Khan. Thank you. A question from Azim Khan. Why are people interested in making a minimal cell? Will it be supposedly closer to the first life form? So our current opinion about that is this minimal cell is derived from a cell that was more complex with a greater number of gene products, and it evolved by reductive evolution. So evolutionarily, it didn't come from a cell with smaller gene products. However, because it contains a minimal number of gene products, some of its physiological processes might not depend on specific and highly evolved proteins, but rather on the intrinsic physical properties of, say, lipid membranes, as considered here. One could also ask, are the intrinsic physical properties of long DNA polymers sufficient to drive something like chromosome segregation? What conformations might one expect for the DNA? And in that sense, these might provide a model system for some earlier cells in which they had fewer gene products, presumably, and these physiological processes were driven by the physical properties of the macromolecules that the cells are made out of. Are you still present, Azim? Do you have any follow-up questions?
Yeah, I want actually what I wanted to ask was like Is it because we are looking for how evolution happened in the first life forms, or is it more like we are checking for the evolutionary aspects? Like, can the degradation in the genomes could lead to a better evolved species or not? What sort of thinking is that we are making in these minimal cells with?
Thank you so much. So just in response to that, I think there's a way in which these cells certainly probably don't mimic some of the earliest cells on Earth, which is they're growing in this complex growth medium where there's bovine serum albumin providing lipid. Uh, and there are a number of people in this audience who are experts on more origins of life and what kind of chemistries and fatty acids and nucleic acids might have been present earlier in the evolution of life. So in that sense, this cell is not a model for those types of processes, but rather the physics of membranes could be studied in the system, and those types of physics might be relevant to those earlier systems. Thanks so much again. Thanks so much for your question. So Angelica Sambor has asked, do you know if those vesicles seen in chemostat contain cytoplasm, ribosomes, etc.? So what we know so far is that those vesicles don't contain constitutively expressed mCherry, which we used as a generic marker for cytoplasmic proteins. We haven't looked at ribosomes. We had two questions. One was, are those membranes, are those vesicles semi-permeable? Are they leaking what those cells had contained? And so we did two experiments there. One, we labeled the membrane with a dye, and the membrane did take up that dye, and so that's consistent with having a membrane. And then secondly, we added fluorescent dextran to the growth medium, which was excluded from those structures over tens of minutes, which over the duration of the experiment. And so in that sense, similar sized structures as a protein didn't permeate into the cell. So they seem to be impermeable. And then furthermore, in some of the dynamics experiments, you can see the vesicles first kind of form and bud off of the surface of a cell. And from the earliest appearance, they don't contain this mCherry marker. So we don't know why they don't contain this mCherry marker. We don't know what else they don't contain. And furthermore, we don't know what's the topology of the membrane in that region. That's a question that we don't know the answer to, and we're curious. So, and actually, it looks like Pete. I'm just going to ask the next question, then come back, Angelica, if you have any follow-up. So, Peter Bandel has asked, are those vesicles a bit like coli mini cells? So, to my knowledge, mini cells do contain cytoplasm. However, there are other structures called outer membrane vesicles, which, to my knowledge, don't contain cytoplasmic markers, but do contain periplasmic proteins. So maybe the vesicles are more similar to these outer membrane vesicles in a sense, but again, we really don't understand the topology of the membrane or how these vesicles are formed, or what the vesicles have to say about regulation of membrane accumulation in these cells. So those are all outstanding questions. Angelica or Peter, do you have any follow-up question? Okay, if you do, please unmute. I'm going to move on to the next question. So Brenton Mortej has asked, this might be a naive question, I'm sure not, but I'll give it a try, and thank you so much. Did anyone ever try, or do you think it would be possible to try, to delete all spontaneous cell division genes for minimal cells and just divide the cells manually by extrusion? That's a fantastic question. I have not tried to divide the cells manually by extrusion. I think it's a very cool experiment. One could also grow the cells in bulk culture and then just rotate the culture perhaps, and then see whether shear forces within that bulk culture, how that affects the cell size distribution. Just qualitatively, I've observed that when I pet these cultures, that it can deform the cells, and although I haven't done that quantitatively, and just as a follow-up point, JCVI Syn 3.0, despite lacking those genes, still does exhibit abnormal cell division where they're constricted shapes. It's not a huge sphere that occupies the entire culture, of course. So, but it's less organized than in JCVI Syn 3A. So I guess a really exciting question would be how might extrusion or shear forces, say, constrain or narrow the cell size distribution? That's something I haven't thought to ask, and think that's a very cool question. So thanks so much. Do you have any follow-up questions, Brendan? Okay. So Azim asked, can you explain what these parameters mean for the cell? Do these parameters affect the rate of cell division? And if so, how? So so that was asked kind of, yeah, so I think the latter part of this talk, we did look into which genes might affect these parameters. And within the context of the spontaneous curvature model, it might be possible to predict the cell size distribution as a function of the relative growth rates of the surface area to volume, as well as the change in spontaneous curvature. And I haven't done those calculations. I'm not sure if the Sfatina in the audience is the Sfatina who's worked on models for vesicles, but there's a 2004 and a 2007 and other papers from Sfatina looking from Sfatina looking at growth of lipids, lipid vesicles, and size distributions that might result from the kind of biophysical processes that could regulate the accumulation of lipid and the osmotic pressure difference driving volume growth. So that work has been done, but I haven't yet done those calculations for possible growth trajectories in Syn 3A, other than what I showed. But I'm interested in that as a future direction. Do you have any follow-up questions on that, Azim?
No.
Okay. Thank you so much for the question. Zane Thornburg, hi Zane, has asked, hi James, do you get an idea of the time from onset of division until the cells are separated from your imaging? Or can you see how long it takes a cell to divide once division has started? So from the imaging that we've done so far, we don't have a sense for those timescales. In particular, the cells are highly sensitive to photo damage. And especially the shapes of the cells was most visible in the fluorescence channel when using that constitutively expressed M-cherry protein. However, we observed that if we image the cells more often than every 30 minutes, then we negatively impacted the growth rate. The cell growth rate decreased. So and then the doubling time is about 105 minutes. So in other words, you can only take 3 or 4 pictures every cell cycle without negatively impacting the growth rate. So it'd be really exciting to look into different kinds of imaging technologies and also trying to engineer the cells to be more stable in response to photo damage, or using other techniques to infer the shapes of the cells over the cell cycle. But yeah, I'm very interested in that question specifically. And I'm wondering, another thing one could imagine is synchronizing the cells somehow and then imaging the cells at different time points post birth. But to date, there is not a method for synchronizing these cells. But that would be an exciting area for future research as well.
It's not too shocking that with such a minimal gene set, it's hard to synchronize the cells. But I completely forgot how photosensitive they are and didn't think about the ramifications of that. 30 minutes is quite a considerable time scale given a 100-minute cell cycle. Yeah.
Seriously, that's why in the movies that we showed that you can kind of get a sense, especially the larger structures, they kind of stay put, and so you can get a sense how they change over a significant amount of time. But we specifically didn't try to analyze shape over the cell cycle because we don't have the data for that. But But it'd be awesome if we could find a way to do that experiment. So really appreciate the question.
On that one.
Thanks so much.
Yeah, thank you, James.
Thank you. And that's the end of the questions in the chat. Are there any other questions from the audience?
James, I have a question that might be stupid, but why are they photosensitive?
I don't know. So mycoplasmas naturally in wild type mycoplasmas, some mycoplasmas naturally have they express hydrogen peroxide, and they kind of use that as an attack against the host. I don't understand that I don't know the first thing about that biology, but based on that, I feel like there should be some genes in the wild type organisms that would confer resistance to oxidative species. But I don't know how those genes act, or which genes might be implicated in that. And yeah, so I don't know, but it'd be cool to try to overexpress things like catalase, or things that proteins that are known to help with photo damage. That's one possibility, but yeah. And then
it's funny that such a minimal genome retained photosensitivity. It's that genome's been minimized to death and back, and yet it's still, that mechanism survived.
I hadn't thought of sensitivity to photo damage as a function. I thought of it more as just a consequence of imaging, but I have to learn more about that. Your questions caused me to think differently about this.
Yeah, James, if I may pitch in just a sentence or two.
Please.
Yeah, no, one of the things with this that even one of the papers James cited, I was the first author on with Syn3A, so I'm quite familiar with the genome at this point. But essentially, one of the big challenges that we were even facing with trying to model such a system is that there's so little regulation left, so little regulation and damage control and damage repair proteins that are coded in the genome. So I'm on James's train here that I also think more of the photosensitivity as a result just of exposure to powerful light sources, and then the cell doesn't have the means to repair itself.
Thanks so much, Zane. Thanks so much. Thanks so much. And then, yeah, Zane mentioned a recent paper as well as previous papers looking at the metabolic models and kinetic models of JCVI Syn3A. So that's beautiful work. And for those who are interested, I really admire Zane's research.
Yes. All right, if there are no more questions, thank you very much again, James, and thanks everyone.
Thank you all. And I'm going to drop my email into the chat for anybody who would like to follow up. Oops, that was a direct reply to Z. Cool. Thank you so much.
Thanks.
Take care, everybody.
Bye, everyone.
Yeah.